Hi, my name is Emily Kodish. It's September 3rd, 2019. Today I will be presenting two case studies. The first one that I will address is how glucose uptake is regulated in the liver, in the brain, skeletal muscle, and adipose tissue. I'll discuss how glucose is uptook in the normal tissue compared to glucose uptake in tumor cells, and I'll also discuss how modifications could have occurred in cancer cells to alter the glucose uptake. Um, so in non-cancerous tissue in the fed state, the glucose is the primary fuel that's oxidized, and the glucose is transported by primary glucose transporters. They're called GLUT. There's 13 different gluc members in the GLUT family, and those 13 members are then further split into three separate classes based on their protein similarities and transport glucose from specific tissue. In the liver, the glucose uptake is regulated by GLUT2 and taken up in an insulin-independent manner. The glucose taken up by GLUT2 will be oxidized through glycolysis and enter the TCA cycle, activate cholesterol synthesis, activate fatty acid synthesis, and the urea cycle. In the skeletal muscle, glucose uptake is regulated by GLUT4 and is taken up in an insulin-dependent manner. When glucose uptake occurs, glycolysis and protein synthesis is activated. Also, glycogen synthesis takes place and the TCA cycle is activated because acetyl-CoA is being oxidized in an aerobic manner. In the brain, glucose uptake is regulated by GLUT1 and GLUT3. It is taken up in an insulin-independent manner, and glycolysis occurs. Lastly, glucose uptake is regulated by GLUT4, and glucose uptake in adipose tissue is regulated by GLUT4, and during this time it oxidizes glucose through glycolysis and stores excess glucose as fatty acids. Glucose uptake in normal tissue is controlled by three hormones: insulin, glucagon, and epinephrine. When glucose concentration is increased in the blood, insulin is released by the pancreas. The insulin then signals the actions of storing the glucose in the liver and muscles where glycogenesis takes place. This turns the glucose into glycogen. However, when the need for glucose comes about, glycogenesis takes place to break down the glycogen into glucose. Every major metabolic pathway in the human body that deals with carbohydrate metabolism participates in glucose conversions, since it's the main sugar in the blood and ultimate fuel for energy in the body. These metabolic pathways range from oxidation of pyruvate, citric acid cycle, pentose phosphate pathway, glycogen metabolism, and gluconeogenesis. In normal cells, the metabolism of glucose has a slight effect on oxygen consumption, such as increased respiration rate, or no effect on oxygen consumption at all. However, glucose uptake by tumor cells had a negative impact on oxygen consumption. It caused a decrease in oxygen consumption in the cancer cells and is referred to as a Crabtree effect. This change in metabolism is actually caused by the quickly dividing cells, and as a result of this respiratory impairment, increased glycolysis. This would be advantageous to the tumor cells because increased rate in glycolysis means a greater amount of energy for the cells to continue reproducing at an increased rate. For tumor cells to produce as much energy as possible in terms of ATP generation, their metabolism is altered. Normal non-cancer cells generate their ATP through oxidative phosphorylation, and these tumor cells shift this ATP generation process from oxidative phosphorylation to ATP formation through glycolysis. Tumor cells will produce ATP through glycolysis no matter how much oxygen is present. Even though glycolysis is a faster process, it actually produces less ATP than oxidative phosphorylation. The next case that I'm going to present is case 4. This has to do with a 2-month-old baby. A previously healthy 2-month-old female presents with jittery spells several hours after breastfeeding. Her mother reports that she's not given the baby anything to eat other than breast milk since birth, and the baby has only recently been sleeping for more than 2 hours between feedings, which is when these symptoms started. Laboratory results are consistent with hypoglycemia. Hypoglycemia is low blood glucose, but otherwise, all the other symptoms are unremarkable. Epinephrine has been administered, and there's no increase in blood glucose, but there is an increase in blood lactate. The baby had a physical exam, which revealed a liver edge 4 centimeters right below the right costal margin. Percussions of the right chest and abdomen confirms hepatomegaly, which is an enlarged liver. Blood glucose levels increase after breastfeeding, but she can't maintain blood glucose levels within a normal range during fasting. So, based on the information that I just expressed, I believe that this baby has glycogen storage disease. I think this is the correct answer because glycogen storage disease is clinically present in infancy or early childhood with symptoms of hypoglycemia, an enlarged liver, and minimal response to epinephrine. These are all characteristics of what the patient has presented in this case. A couple of additional symptoms of this disease are hyperlipidemia and growth retardation. This disease is not common and it is predominantly seen in children. It's congenital and cannot be acquired from other environments and the body stores glucose in an alternative way. This is because the infant's body is missing an enzyme that's involved in breaking down glycogen, and since it's missing, this causes glycogen to build up in the liver or even cause the glycogen to not form the right way. There's actually several different kinds of glycogen storage diseases, and each type is organized by the enzyme that it is lacking. All the glycogen storage diseases are different and have different symptoms from one another, so they all require different treatments. Now I will discuss why the other diseases are not possible diagnoses. So galactosemia is a hereditary metabolic disorder. It's an autosomal recessive inherited disorder of carbohydrate metabolism. In this case, it's the incorrect answer because the primary symptoms in newborns for this disease are vomiting, jaundice, sepsis, full fontanelles, and hepatic failure. None of these symptoms were presented in this case, and therefore galactosemia cannot be the correct answer. The next incorrect answer is hereditary fructose intolerance. It's incorrect because fructose is principally metabolized in the liver, and the initial step in this pathway is to phosphorylate the fructose to the 1 position with fructokinase into fructose 1-phosphate. The second step is the rate-limiting step for fructose metabolism, and the enzyme aldolase B is used to cleave the fructose 1-phosphate into dihydroxyacetone phosphate and glyceraldehyde. In the case of hereditary fructose intolerance, there is a deficiency in aldolase B. So the result is an accumulation of fructose 1-phosphate. The baby would not have hereditary fructose intolerance because in its case it states that hypoglycemia is present in the fasted state, but in hereditary fructose intolerance, hypoglycemia is actually present in the fed state. And lastly, glucose 6-phosphate dehydrogenase deficiency is not a probable answer either because it's found almost always in males. The patient presented in this case is female. Also, symptoms of glucose 6-phosphate dehydrogenase deficiency are paleness, jaundice, dark urine, fatigue, and rapid heart rate. None of these symptoms are present in this case, and that helps us rule out this deficiency as a probable diagnosis.